my pleasure to introduce Sam Scarpino. Sam got his bachelor in biology at Indiana University, and although I was at Indiana University, we never met there, and I'm much older, but I was there. And then he moved to have a PhD in ecology and evolution and behavior with Professor Meyers at the University of Texas, Austin. Then he has been a postdoctoral fellow at the Santa Fe Institute, and he was one of those lucky researchers to get the OMIDR fellowship. It is a very prestigious fellowship that gives you complete freedom to explore many topics, and he did a great job at Santa Fe at starting to work on several fronts. And then he moved as an assistant professor in the Department of Mathematics and Statistics at University of Vermont. You see that's a very interdisciplinary background from biology to mathematics, statistics, ecology. And today he will talk about the predictability of outbreaks. Please.
Thank you. Thanks, Alex, for the introduction. Thanks, everybody, for being here. I actually realized that I don't think I've given a talk to so many network scientists before, so I actually probably stole this Juan Genovese from Spain, from maybe one of you in the room, because I saw it at a talk, I think, at NetSci last year. This sort of creates these network things. So if it was your talk, I apologize for not crediting you appropriately. So I'm going to talk about the predictability of outbreaks, but what I want to begin with are sort of two much shorter vignettes that led us to think about this question about predictability. And the first one comes from work that we did during the Ebola outbreak. And one of the really amazing things that happened during the Ebola outbreak was how much data sharing occurred amongst the international community of people that were working on this problem. Now, we of course still have a long ways to go, but one of the things that happened is that in July of 2014, this data set was made available on GenBank. So what we're looking at is a phylogenetic tree of about 100 Ebola virus genomes. So each tip on this tree is a genome of an Ebola virus that was isolated from a patient and sequenced. So if we want to think about how closely related these sequences are, you would say start at the tip and walk back down and then move back out to the next tip, and that difference would tell you how close those two genomes are to each other. Now, one of the things that we can do from this tree is we could try to infer some of the parameters of the spreading process directly from, say, the branching rate of this tree, right? So the amount of genetic diversity in the genomes of these viruses is going to be affected by how many people they're moving through, how fast they're moving through, of course, the mutation rate, and those sorts of other things. You can, perhaps if you stare right away, you can see there are these two deep branches, and we know what happened is that two different individuals were infected at the same funeral, went to different parts of the country and started two transmission chains. So even without any modeling at all, we can learn something about the immunological process. But what we wanted to do to start with was to, instead of look at this as a phylogenetic tree, we wanted to try to infer the network of who infected whom from these data. So in the paper, they say that they think they got 80 to 90% of all the individuals that were infected and showing symptoms with Ebola during this time frame. So we basically have the entire transmission chain sitting in front of us. And we know something about the probability of mutations arising in the Ebola virus. We think we know something about how those mutations get fixed as they bottleneck through different individuals. And so we can write down a model that tries to wire up the network of who infected whom. So now the circles are individuals. The size is proportional to the number of individuals we think they infected. And the color tells us when we think that they got exposed to the virus. So one of the hallmarks of Ebola is they can have this very long latent period of infection between exposure and symptoms. And again, it's a little bit harder to see, but you can kind of see the two transmission chains here. One of the other things we're able to do is you see all these disconnected nodes. We're able to try to independently estimate the reporting rate by saying how many links are missing, how many nodes are missing, and try and solve that problem both from a genetic perspective but also from sort of a network science perspective. And we can ask another question, which is one of the key parameters that we use to compare models that we use to make public health decisions is the number of secondary infections. It tells us the growth rate of the virus. It tells us how hard it's going to be to control. But of course, if you've worked on this before, one of the issues with this number of secondary infections is that of course it's going to vary based on the social network structure based on the virus, but it's also going to vary just for all sorts of other reasons due to stochasticity. And often this one single parameter describes a heavy tail distribution. So instead of fitting a model and estimating the growth rate, I can just ask the data how many secondary infections each person had. And we of course don't have a single tree here, nor do we have a single tree here. We have a posterior distribution over trees and over transmission chains, and so we could construct a posterior distribution over secondary infections. So what we're looking at is the posterior distributions of the number of secondary infections. And what you see is what we know, or what we think we knew about Ebola and turned out to be true, is that the sort of mean of this distribution is bigger than 1, so we're going to expect kind of exponential spreading in a lot of populations, but it's bigger than 1 because it has this very long tail. So we have some individuals in this population that are giving it to 12, 15, 20 different individuals. And so from a control perspective, that tells us something about what we might do to intervene, which is that if we could bring down the upper end of that distribution, say by protecting individuals that are dealing with funeral, with bodies that are in healthcare settings, then we can have a really big effect on the number of infections because we're just right over that border in terms of the number of cases. But the other thing that we wanted to think about is that for Ebola, the types of social networks or the links that the disease is going to be traveling over, especially early in the outbreak, are likely to be special. Special with respect to what we might do if we just looked at the social network for Boston and calculated a bunch of metrics on it. And special because we know it moves through households and we know it moves through workplaces. Those are the individuals that get hit disproportionately hard at the beginning of these Ebola outbreaks. And individuals that are sitting in that same household, sitting in the same workplace, are going to be in much more densely clustered networks than in individuals in the larger social network. So this is a picture here of the father of patients, what we think are patient 0 in one of the outbreaks, standing in front of the tree that they think maybe the bats were sitting in that infected the 2 patients. And clustering is something that we actually know a little bit about in terms of how it affects the process of something spreading through a graph. So if we have 2 different networks with the exact same degree distribution, but one of them has a higher clustering coefficient. In this case, I actually mean the clustering coefficient, about the fraction of closed triangles. So we have 2 different graphs here. This has high clustering, low clustering. It's actually the hardest part of this talk and the research project. They do have the same degree distributions, which is really not easy to draw 2 different graphs. They're this small that have the same degree distribution. And I'll just play the game where I'll infect the red node, and then with probability 1, every time I click the mouse, I'll infect all of their neighbors. And at the end of time, there's still 2 individuals that are not infected here and 1 individual that are not infected. And this turns out to be a mathematical fact that on a graph with the same degree distribution with different clustering coefficients, you'll have slower propagation through the more clustered graph, right? And that's because friends of friends create these sort of blocks in terms of the propagation, which are going to slow down the rate at which the disease spreads. Interestingly, you can actually still end up having the same average number of secondary infections, even though one of them is moving faster through the graph. The way that you would have to tell that apart has to do with the variance in the degree distribution. So what we wanted to ask is, do we see this effect in the pace and tempo of Ebola cases coming into the population? So we posit that Ebola is moving through more clustered social networks. The math tells us that it should be spreading more slowly. Can we actually see this in the data? So what we did, and I probably can't actually see the data in the back because I can barely see it here, but so these are the cumulative number of cases in Sierra Leone from May 27th through August 30th. And we're going to fit 2 models to these data. Both models are social network models with parameters taken from household survey data in Guinea during the outbreak. One of the models has no clustering in it at all, so no closed triangles. The other will have some non-zero fraction of closed triangles that we will try to estimate. So we'll fit both of these models to the data. And so this is the best fit model without clustering, and it's a little bit hard to see. So this blue is fit to the number of cases, the incidence, and the black here is fit to the mortality. That both the blue and the black are significantly up above the data at the beginning of time. So what the non-clustered model is doing is it's predicting too many cases, too high of a rate of infections coming into the population, especially early on. If we then fit and allow the clustering coefficient not to be 0, we get a much better fit throughout the whole curve, but in particular we're getting this early pace and tempo of cases correct. Now interestingly, the clustering coefficient that we estimate is absurdly high. It's 0.7. But given that these are households and workplaces, that's probably not completely out of the realm of possibility. Now, one of the interesting things that ended up happening is that the reviewers said okay, we believe you that you tried really hard to control for this extra degree of freedom in the model, right? So this model is more complicated than the other model, and so maybe we're not so impressed that it fits a little bit better because you have this other thing to play around with. You already have a lot of parameters to begin with. And we think we tried really hard to deal with this, but they said, here's another way we can test it, is that it's been 2 months since you sent us the paper. Just don't change anything, turn the computer back on, tell us how many Ebola cases there are going to be. And then if you're close, we'll publish the paper. Right? So no pressure. So here's the proportion error on this 2-month out prediction. So we fit through August 30th, and then we project out to November 2nd, and we just compare the number of total cases. So this would be a 20% under prediction, this would be a 30% over prediction. This is for our best fit clustered model. This is for our best fit unclustered model. So our best fit model under predicts the number of cases by 15%. I'll say more about that in just a second. Now you might ask, did you try to minimize this out of sample error, right? So I might then try to refit my model a bunch of times such that I get this thing to be 0. And what clustering coefficient do I get? Well, the clustering coefficient I get is 0.3, which matches the clustering coefficient in the social network that they got from the contact tracing data, which matches a lot of other published estimates of the clustering coefficient from large social network data. So what we think is happening is that as the disease spreads, it moves out of the household, it moves out of the workplace into the general population, which is going to make it both harder to identify secondary contacts because they're going to be maybe more distant in the social network, but we would also expect the pace and tempo of cases potentially to pick up because it's realizing a lower clustering coefficient. The second thing is that at least from these data, there's no evidence of interventions. Now what I don't mean to imply is that the interventions weren't working, but what I mean is that I can take a still relatively complex but fairly simple network model with clustering, but no interventions, and underpredict the number of cases that we would end up seeing through November. So what this told us is that if we were going to look for the effective interventions, we would need to do it either at a different spatial scale or with different kinds of data. And that's one of the features that we've been seeing a lot of and that a number of people here have worked on is that the types of data that we have in these different social systems, these different biological systems or technical systems, give us different kinds of information about the processes that we're interested in. The third is that perhaps we can forecast outbreaks. So there was a lot of press around the forecasting of Ebola, in particular because of this CDC forecast. So this is the CDC's forecast paper, and if we do the same 2-month out prediction, there were 10,000 observed cases and the CDC predicted 100,000. So they were way off. We were under by 15%. And we think this is because, so they fit a mass action random mixing model to the data and then ran that model forward in time. There's a whole number of reasons we might imagine that process to break, right? So what this is kind of telling us is that the model matters, right? That capturing some of the relevant or most relevant features of the system is going to matter in terms of generating good forecasts. The next thing that we started thinking about from a network perspective is what happens if this healthcare worker gets sick from Ebola during the outbreak? Well, what happened during Ebola and what happens during all outbreaks is that that person gets replaced. A new healthcare worker who is healthy will come in to fill the role of that individual. And this happens a lot. So school teachers get replaced if they get sick. Individuals who are involved in all sorts of societal roles that are essential, police, firefighters, etc., they get replaced if they get sick. And what this replacement does is in a social network is perturb the relationship amongst individuals. And we wanted to get a sense for what kind of dynamics this might induce on the system. So what happens if we replace sick healthcare workers? So we have a network here. Edges are indicating places where disease can spread. Red are infected, blue are susceptible, and maybe this is a nurse caring for these 3 individuals. And if they get infected, they get replaced by a healthy individual from somewhere else in the network. We want to think about what this does to the dynamics of the spreading process. And it turns out that if you look through the literature, there is lots of evidence that we replace people all the time. So school teachers, on average, will miss 3 days a year due to illness. There was this really interesting study in Maine at a high school where they didn't vaccinate the teachers. They only vaccinated the students against influenza, and teacher absenteeism dropped by 10% over the course of the year due to illness, which is pretty stunning given that they didn't vaccinate the teachers and they have all these social contacts outside of the school. we know during the 2009 H1N1 pandemic that lots and lots of nurses got sick and had to be replaced by healthy nurses. And then even sort of not in Vermont or in Canada, but like seemingly silly things, but still have the same network effect. If individuals on a sports team, for example, get sick, and you have to move somebody from the reserve team onto the main team, that's going to perturb the social contact patterns. So this is work that I did with Laurent Dufresne and Antoine Allard. And it turns out that what happens is pretty fascinating. So if we have a mass action model, a random mixing model, so the way that I decide who to have lunch with is I just grab people at random from Boston and I have lunch with them. If I shuffle the labels, that's not really going to do anything interesting, right? Because everybody's already mixing random with each other already. Right? Instead, once we induce a network structure, we start to see something really fascinating. So this is a susceptible, infectious, susceptible model. So what that means is that I'm susceptible, I can be infected. I can then become infectious, meaning I can transmit the infection. And then after some period of time, I will recover, but instead of being immune, I will go back into the susceptible population. So this is the proportion infectious at equilibrium. Sorry, this is the proportion infectious over time, not equilibrium. Proportion infectious over time for a standard SIS model on a network. And what you see happening is that you have exponential growth. This is on a log scale. So you have exponential growth. And then we're going to have relaxation into some steady state. So you're basically going to be at this constant fraction of infectious people for all time. This black curve is showing us the exponential growth phase. Now we're going to start replacing individuals that are sick. So that become sick who have key societal roles. So what we'll do is we'll just tag a fraction of the population as being essential, and then we'll have some rate parameter that says if these essential people get sick, how fast do we replace them? And we have this rate parameter because how fast we replace them relative to the recovery rate, relative to the force of infection matters in terms of what types of behavior we see. So the blue curve is what happens when we start replacing people. And we have exponential growth early on, and then we have exponential growth later, but at a significantly higher rate than the exponential growth we saw in the standard model. And then when we get relaxation into the endemic part of the curve, it's always higher than without replacement, right? So what's happening here is not particularly surprising, right? So what we're doing is we're basically inflating the number of susceptible infectious edges in the network, which is the fuel for an epidemic process spreading on a graph. A couple of other really fascinating things happen though, which is if we look at the proportion infectious at equilibrium as a function of the rate at which I replace these individuals relative to the rate at which they recover and go back into the susceptible population, we see this hysteresis loop, which is that if the rate of replacement is fast enough, then we don't have individuals who are infected at equilibrium. So if we replace people fast enough, we can keep the outbreak from spreading. If we replace people too slowly, then we end up with this non-zero number of infectious individuals. But let's say initially we're really on top of it, or we have a large pool of individuals we can replace from, and then we either become complacent or we run out of individuals we can replace with, and so we back off on this replacement rate. Well, what it does is it drives us over, I'll show you in a second, this discontinuous transition into the endemic phase, and then we have to crank the rate of replacement way up again in order to drive the system back into the non-zero endemic area. And so you actually probably can't see this, so I'll just tell you what it says. So this is the proportion infectious at equilibrium, again as a function of the infection rate divided by the recovery rate. So this is the force of infection divided by the rate at which they recover for 3 different increasing rates of replacement. So this is slow replacement, fast replacement, really fast replacement in dark blue. And what you can see for the 2 lighter colors is that the transition into the endemic phase is continuous. So it's 0 and then continuously increasing for both of these. But for the dark blue, it's a discontinuous transition in the endemic phase. So you have 0, 0, 0, and then you jump all the way up to 0.5. And we, of course, the real truth of how we figured this out, right, is the opposite, right? We saw this, and then we assumed this had to be there, so we found it. We're telling you in the opposite direction. Now, you might kind of wonder, is this just an artifact? So these are not simulations. These are the actual solution to the system of equations, assuming that everybody has the same degree. And so you might wonder, is there something funny with that sort of approximation? You also might wonder that a lot of other things could happen, is that I could come into a replacement role and then I might go back to my original role and then that person would come in. Maybe I move some of my social links with me. So we relaxed all of these assumptions in agent-based simulations, and what you can see, this is the time evolution proportion infectious on a log scale for a whole bunch of simulations, and these big jumps here are this exponential speed up that we're seeing in terms of the spreading, and as best as we can tell, it isn't any of the assumptions that go into the model that are giving us this exponential spread. We don't see it sometimes, just we think due to stochasticity in terms of the simulation. So there isn't any of those assumptions in particular that leads to sort of a pathological decay of our results. We then wanted to ask whether there's any evidence for this in data. And the way that we wanted to ask this was to look for this exponential speed up. Most of the existing models for disease transmission, they don't really allow for this kind of speed up right towards the peak. So if you work on influenza, there are a lot of seasonal forcing models, but usually what those do is they change the rate of infection to kick off the outbreak. But the actual force of infection is constant across the peak. And so what we wanted to do is look for this accelerating spread, because it's the kind of thing that we think is not really very common. And we're going to compare influenza to dengue. And the reason we're going to compare it to dengue is not because individuals with dengue don't stay home sick. They probably stay home sick at a higher rate because it tends to be, at least on the first couple of infections, more severe than influenza. But because there's this separation of timescales between the transmission process and the behavioral process. So for dengue, the mosquito has to bite me. It has to replicate out of the gut of the mosquito into the salivary glands, and then fly over and bite Alex, who also has to go through a latent period before he would have to stay home. So we think this separation of timescales is going to mean that we won't see this effect in dengue. So the way that we did this, we did this as sort of fully extreme way, which is that we fit two different models, one with this relational exchange rate being non-zero, one without, and we have statistical support for this relational exchange rate being a better model and a non-zero rate. But we actually think, or at least my co-authors and I think, that this is a more compelling result, which is we took historical influenza data in the US and we fit exponential growth to the first 8 weeks of the curve. And then we ask if the second 8 weeks of the curve is statistically higher with respect to its exponential rate or not. And if it is, we color it in red. And again, we have the same results doing this sort of model fitting exercise as well. So this is data from 1998 through 2014. And we see this accelerating spread leading to the peak of influenza every single season, all three waves of the 2009 pandemic. For dengue, we do the same exercise, and we see it once or twice over the entire time series, depending on how we set the signal. So we really don't see it in dengue at all. Now, one of the things that we're actively working on is how to evaluate this more rigorously, but it turns out the problem is in the parameters. So the key parameter here is the rate at which people get replaced, not the fraction that get replaced. And so there are a lot of papers that have been published on the fraction of nurses that get replaced or the fraction of teachers that get replaced, but not how fast they get replaced relative to the onset of the disease. So we're trying to think about how we might actually be able to gather data on that rate of replacement to kind of evaluate this more fully. There are, of course, a number of other possible behavioral models that could lead to this kind of accelerating spread. We think that that's going to be a really rich area of future research to think about how different behavioral processes, when they're then coupled with a spreading process, can lead to different kinds of nonlinear dynamics that we maybe wouldn't see with just that spreading process. And this led us to kind of start thinking about these issues of predictions, right? So we've seen that the data can matter a lot in terms of the quality of the prediction, that the models can matter a lot in terms of the quality of the predictions. And we want to try to then study the sort of physics of prediction, or sort of the information theoretic properties associated with some of these time series that we want to do prediction on. And we, of course, care a great deal about this. So this is a map of H5N1 bird flu in domesticated bird populations. There's been a lot of news around H7N9 right now because the cases are increasing. So we care a lot about making these predictions in terms of the emergence of pathogens after they emerge, where are they going to spread, how many cases they're going to be. These things really matter. They've had a big effect on outbreaks going all the way back to SARS, Ebola, H1N1. All of these forecasts have mattered in terms of the international response. they also matter in terms of how we do science, right? So one of the ways in which I would compete my model against Alex's model is we would see which one performs better at cross-validation, right? So we would maybe train our models on the blue parts of the curve, and then we would evaluate them on held out data, the red parts of the curve. So the ability of our models to do prediction is sort of, depending on who you ask, actually the scientific method, right? This sort of cross-validation for prediction. And so Giovanni Petri and I at ISI are kind of trying to ask this question about the information theoretic properties associated with predicting outbreaks. And the way that we want to try to answer this question, which is can we predict this time series, or how fast does this time series generate information, is in a model independent and a metric independent way. Now this has some costs because what we can't tell you is whether a particular prediction you're interested in can be made or not. So making a prediction requires a model and it requires some metric, right? So we have to say that we're gonna fit an SIR model with this degree distribution and these other parameters, and we're gonna evaluate the models using the root mean squared error or the log likelihood or something like that. Okay? And then we can tell you whether this prediction is better than this other prediction. But what we want to try to avoid doing is arguing about whether we have the right model or not. And also avoid arguing about whether we have the right metric or not. So one of my favorite conversations of all time, it actually happened while I was eating a hamburger in a car. I had a computer scientist and a statistician who were working on a project together on conference call, and they're screaming at each other about whether we should be using the out-of-sample log likelihood or the out-of-sample root mean squared error to evaluate our model. Of course, what we ended up doing was showing they both gave us exactly the same result, but they both had deeply principled reasons why they had to use this particular metric. And so we want to really avoid that kind of argument. So what we're going to do is we're going to focus on a quantity called the permutation entropy. And the permutation entropy tells us in a model and metric independent way how fast is the system generating information, how predictable is this system. In 2 dimensions it's relatively simple, so if we want to predict this time series, or say something about the predictability of this time series, in 2 dimensions they're just 0 and 1s, we count up how many 0, 1s, 1, 0, 0, 1, 1, 1, 0, 0 we see. And we build a distribution over the occurrence of these different symbols. And the flatness of this distribution, or the Shannon entropy, is the permutation entropy of this time series. Now in higher dimensions, we could create symbols in other ways. So we could say, have a time series that looks more like an influenza outbreak, and maybe we have down, 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 or down, flat, flat, or up, up, up. So we can define these types of symbols. We have to pick the dimension that we're going to look for. And the way that we do that in practice is we just sweep over a huge number of dimensions and find the one in which we can do the best kinds of prediction. And I'll say this at the end, but this is actually one of the things that we think is going to be most useful about this approach, is that if you give me a big data set, we can say something with permutation entropy about in which dimension is it going to be most predictable. And so we can look at the data and say, actually, if you kind of coarse-grained it by weeks or fine-grained it out to hours, we actually would suggest that you could improve the predictive accuracy of your models. And there was a very nice paper by Joshua Garland, who was doing a postdoc with Liz Bradley, that they just published in PRE, showing that the permutation entropy correlates with the prediction error for a whole bunch of models that we know have different limits to their ability to do prediction, right? So if I give you a chaotic time series, I can say something mathematical about my ability to do prediction with existing tools, like Lyapunov stability type tools, and so I know what I should be able to do. And so what they were able to show with this simulation is that the permutation entropy does tell us something real about doing actual prediction. Now, of course, it's complicated, right? So I mean, it's definitely a nonlinear function in terms of how it's related, so the interpretation is very much going to be context dependent. So what we then wanted to ask is, what does the permutation entropy of this time series look like? And there's a couple of ways in which we could go about doing that. One is we could kind of play the Bayesian game, which is we just start looking at longer and longer chunks of time series and calculate what the permutation entropy looks like. Now, if I do this for a sine wave, that permutation entropy will go down, and then once I've gotten through a full phase of that wave, it will be flat. If I do this for white noise, as long as I've seen at least d factorial time steps, where d is the dimension, it will just be flat and at 1. If I do this for white noise. Similarly with a chaotic time series, as long as I get the dimensionality right and I've seen enough of the data, it will just be flat and basically at 1. So here's what it looks like for this influenza curve. So this is the number of years of data that we've seen versus the permutation entropy. And there's a couple of take-homes here. So one of them is a positive message, which is, so if I do this for a sine wave with a little bit of noise in it, I get a value of around 0.8. So we sort of see 0.8 as being kind of a key value where predictability might start to become hard. Is that through the first couple years of influenza, it looks like we can do good prediction. So the predictability goes down as the entropy goes up, but at least through the characteristic timescales of flu outbreaks, there's a lot of information that's present that we can use to do prediction. The second thing, and depending on your background, this may be more or less surprising, is that it goes up. And it's not going up because we haven't seen enough data. It's going up because the actual stable distribution over those symbols changes in time. And in a Bayesian world where the likelihood equation is fixed, this is not allowed to happen. Okay, the entropy has to go down. The KL divergence of the posterior has to get smaller. In this case, the KL divergence blows up between these different distributions as we see more and more data. Just for sort of some help here, this is a sine wave with really no noise, and it will actually eventually go down. There, you had to add a little bit of noise to get this to behave itself. And then this is a sine wave with a lot of noise added to it, although still very predictable through the first couple periods there. So we looked at this at a bunch of different influenza time series data. This is the permutation entropy as a function of the length of the time series in weeks. And we see this continual increase. As we see longer and longer time scales, the entropy goes up and up and up. Now it eventually gets to 1 and sticks there. But what this tells us is that the longer the time series we have, the higher the entropy is going to be. And this says something about the way we should be thinking about prediction, which is not really news to a lot of you here, but the dynamical approach to forecasting, where we're training models, updating models, generating forecasts, is very likely going to work in these systems and likely be far superior to systems that are relying on long historical time series to train model, because the model structure really appears to be shifting. The other thing that this seems to tell us is that if Alex and I want to duke it out over whose model is correct, running a whole bunch of cross-validations on different influenza seasons may or may not be that informative about which one of our models was right in any one particular season. Because he could be right in some seasons and I could be right in other seasons, and we may or may not be able to tell that by running a bunch of cross-validations. We also looked at a whole bunch of other diseases just to see that this wasn't an influenza thing. And so what we're looking at is over the first 2 years, so here is 1 year, here's 2 years. Over the first 2 years for Zika, influenza, polio, measles, whooping cough, mumps, gonorrhea, hep A, and chlamydia, we see this, so this is now 1 minus the permutation entropy, so this is the predictability going down. We see this happening for all these different diseases. But again, the characteristic timescale for most of these diseases, at least in the US, is about a year. And so they all are very predictable through sort of one phase of their outbreak, which is kind of a reassuring message. What we're actually looking at in this case is I have 1,500 time series, and for each time series I pick a random starting point, and I do this 1,000 times for each time series, and I then calculate out the permutation entropy at 1, 2, 3, all the way out to 100 weeks, and this is the median value for all these different diseases. So there's going to be a big spread around all of these depending on exactly where in the time series I pick and exactly where the outbreak is occurring. So for example, influenza in a small area is much harder to predict than influenza in a big area. If we zoom out a little bit, this is over much longer time scales, and each color is for each different disease. And there's a remarkable amount of clustering by disease. There isn't any particular reason why we should have expected this, but all of the chlamydia outbreaks are together, all of the flu outbreaks are together, all of the whooping cough and measles outbreaks, the polio outbreaks. And if you work on disease and you're kind of staring at this a little bit, you might be tempted to say, is this structured by differences in the number of secondary infections or differences in the social number? And we think the answer is yes. So these are going to have a much higher first moment in the number of secondary infections. Measles, polio, whooping cough, they would infect everybody here if you're susceptible to them. Whereas for influenza, it's going to be much closer to 1. at least for seasonal flu, the effective number of secondary infections would be closer to 1. And chlamydia is kind of interesting because that's a sexually transmitted disease, and so maybe we don't know as much about the first moment, but we do know something about the second moment, which is it's probably moving on a power law distribution. And so there is also some evidence that maybe the second moment of this number of secondary infections matters. So we're going to zoom way out, and I've renormalized time by the average or the most reported reproductive number I found in the literature. We're on a log-log scale here. And the slope of this line is 1, and I can predict the differences in this slope in a random effect model using some information that I think is telling us about the second moment of the distribution, which is this embedding dimension that's left over. So we wanted to try to push on this idea a little bit, which is, do the characteristic differences in the social network that maybe lead to differences in the number of secondary infections, do they tell us something about the permutation entropy or the predictability of these different systems? And the answer appears to be overwhelmingly yes. So what we did is simulated a bunch of outbreaks on social networks with different first and second and higher moments of their degree distribution, which then lead to different moments in terms of the number of secondary infections. And we estimate the permutation entropy from all these outbreaks. I'll go back to that in a second. So we have high heterogeneity, sort of intermediate amount of heterogeneity. So this would be like maybe undefined second moment, high second moment, almost a lattice in terms of the degree distribution. And then when we set the transmissibility such that these basically give us back what the number of secondary infections look like. And here's the resulting permutation entropy for these time series. So what we're actually looking at on the bottom, if you're familiar with spreading processing on graphs, there's this quantity called the critical transmissibility, which is what is the per contact probability of transmission, such that in an infinitely large graph, we're going to expect to have this giant component emerge of infected individuals. So this is the distance that this system is from criticality. So this thing is sputtering chains far from criticality. This thing is basically giving us nearly deterministic outbreaks. And what we see is very high predictability here, and much, much lower predictability with a big spread. And of course, this spread is predicted if you just go in and look at whether this thing is generating a curve versus this, that predicts what these spreading differences look like. And so we see this very strong effect that maps on to what we saw in terms of the diseases. So the diseases with the sort of lower first moment, higher second moment that you're going to have more stochasticity tend to have lower predictability than the diseases that, like measles, that are generating these very regular, although they're chaotic in some places, but very regular waves once they start to occur. The other nice thing about this is you'll see that the permutation entropy for all this is much lower, and that should be the case. This is a relatively simple model that we're simulating here, so the predictability should be lower. Sorry, the predictability should be higher, the permutation should be lower for all of these systems. The last thing I'll say about this is we see some other evidence for this kind of shift in the number of secondary infections looking at the natural experiment of vaccination. So this is measles data in the US from 1927, this is Texas, through 1983. These are the cases, this is the permutation entropy. This is when we introduced the vaccine, and we see a significant rise in the permutation entropy after the introduction of the vaccine. That's what we would predict if we have a reduction in the first moment of the number of secondary infections, which is what is supposed to happen with vaccination, is that you reduce the opportunity for secondary infections. And we see this sort of permutation entropy start to climb in the way in which we would anticipate. Now, one of the things that may not be immediately obvious about the permutation entropy is that it's going to be, and this is a bit of a challenge, say, if we wanted to do this for actual forecasting, but it's beautiful for this, is that it's robust to all sorts of monotonic transformations of the data. So this increase in permutation is not just because I've shrunk the curve, it's actually because the patterns in the curve have changed. It's going to be robust to those changes. So what we're trying to build up here is this complex systems integrative biology approach to thinking about disease. And I didn't talk about this, but I have ongoing research involving how differences in socioeconomics in terms of access to healthcare, health-seeking behavior affect the pace and tempo of cases coming into the population, affect our situational awareness because we get our surveillance data from primary healthcare providers that individuals in low socioeconomic groups don't have access to. Things like airline travel networks are going to affect the movement of disease, so we know networks matter a great deal. We know that the networks probably differ in different populations by socioeconomic status, maybe, although that's, I think, an interesting area of open research, how those differences might be structured. Human behavior change is going to have a big effect on these spreading dynamics. If we start sending individuals in blue suits onto airplanes like we did with Ebola, that's going to change the way in which individuals are going to react to these diseases. We also know that the evolutionary dynamics, in terms of our ability to both do inference, but also how this thing might respond to drugs or vaccines, or how this might affect the predictability or the pace and tempo or the virulence of new infections that might emerge, all of these things are going to interact with each other. And that, of course, creates a big challenge that all of us are embracing, which is that we have to take this integrative approach to modeling. We have to lean on each other's expertise because we don't have the ability to become experts in all of these different areas. It's why we need the kinds of collaborative groups that you all are building here. But it also creates this really exciting statistical opportunity, which is that, okay, these things are connected and we've got to build these big models and it's a really hard procedure, but the evolutionary dynamics tell us something about the behavioral dynamics. There's a pattern that's there. The behavioral dynamics tell us something about maybe subpopulation or movement. All these things are linked, which means the data sets are linked, which means that we can leverage them to improve our ability to evaluate our models, to do prediction, to learn things about these socio-biological or maybe socio-technical systems. With that, I'd like to thank Giovanni, who's working on the prediction limit stuff with me, Laurent and Antoine on the relational exchange. Ebola had a large group. Achal and I were the co-first authors on that paper. And then funding from the NIH Midas group, the Santa Fe Institute, the Omidyar group, the University of Vermont. Happy to take questions. Thank you.
The first question is about the Ebola and the influenza for the surveillance data. So do you expect at the beginning there will be any reporting?
For Ebola?
For Ebola or influenza?
So for Ebola, we know that there was a problem with reporting early on. So very early on, it was being misclassified as a different disease. The thing with Ebola that I'm most interested in is, so I didn't mention this at all, but if I'm a population geneticist, which I was at some point and still have the hat in my suitcase, I can write down a probability distribution over how I expect these trees to bifurcate. And if I have random censorship, random unreporting, I can still get an unbiased estimate of the growth rate. So if I just randomly censor some of the tips. But for Ebola, the tips probably aren't randomly censored. So if you don't get reported, that's going to change the probability that I don't get reported if we're in the same transmission chain. And that thing makes things much more complicated. So for Ebola, I think, and this may be maybe me just being naive, I think it's not so much the underreporting, it's how the underreporting is happening. Whereas for flu, we definitely know that there's underreporting early in the season because physicians are sending in the data, and physicians seem to have this behavioral filter where until they see a lot of flu cases, they don't start saying it's influenza. So I think maybe for flu, it's probably random but much more pervasive.
So for the flu, then the strategy you did is like feed the first 8 weeks. So that would be kind of like you could underestimate.
Yeah,
and then that's the thing, and then you see the speed up. I'm not sure it's because of underreporting or is actually.
I agree with you. So the question, which is a good one, is if I change my reporting rate, can I get a speed up? And I think it's absolutely, the answer is yes. And I think that is also an equally likely explanation for the data that we've shown. As far as I know, there hasn't been studies that have been done on this. There may, and I'm probably really stepping on somebody's toes now, so please tell me that I'm missing this. So what I know, the CDC did a study where they tested every individual that was classified as ILI, having influenza-like illness, for a battery of respiratory infections. It's called the Influenza Incident Surveillance Program. And the first year of that study, you see this reporting bias in the physicians, and then it goes away in the second year. And we think it goes away in the second year because the physicians saw the data and they saw they were screwing it up and they adjusted how they were applying the case definition. So it's really, I would bet heavily that that's what's going on. We actually, the first iteration of the paper we wrote was, it's a long-standing question why we see this speed up in the influenza-like illness data, but we couldn't find citations on it. So yeah, it's,
so like when you design a model, do you see the speed up first and then you come up with an explanation, or you come up with the explanation first and then?
so you're asking me to be honest about the order.
Yeah.
turn the video off. No, so we came up with a model based on the Ebola outbreak. So we were interested in what happens if we replace sick healthcare workers. And one of the things that we have found is that we can really kind of strengthen the appeal of our modeling studies if there's some way we can try to ground this in data. And so this was our attempt at showing that this kind of process may be happening in the real world. And admittedly, we hope we were very forthcoming with this in the paper, that this is very preliminary. And we actually think that the more robust way to do this is to try to go out and directly measure this replacement rate, or do the kind of behavioral studies that you're talking about.
Excellent talk. Did you look at multi-scale prediction entropy in your work?
Yes and no. So what we did look at was how the permutation entropy changed over the scale at which we were looking at the data. We also looked at how, if I say were to combine data, like let's say that I put all of my influenza time series in a matrix and I do like a singular value decomposition and take the loading onto the singular values as a new time series, we looked at how the permutation entropy scales with that. And we can definitely find different embeddings that have better predictability, if that's what you're asking. But maybe that wasn't the question.
No, but that was a very good answer.
But not the question you're asking.
So the question is, if I actually have a tree, like a multi-scale, and in each one I can see at what level of the tree do I need to be to do better prediction,
Yeah.
right?
We didn't look at it exactly in that way, but you certainly could. And we certainly, I mean, we absolutely see that depending on the granularity of the data, we have really different abilities to do prediction. And so I think that would be a really fruitful area to look into.
So I think Tim is asking how that scale was selected.
Well, how was the scale selected? If you're asking how, we just,
No,
sorry,
I was-
if you, we'll wait, to answer that question, we just looked at all possible scales modulo the computation time and picked the one that gave us the lowest entropy.
Right, but actually the thing that I was asking was what physicists say multi-scale,
Yeah,
right? Like you actually have this tree and so on and so forth.
we didn't look at that. We
I
should,
see.
which is,
Okay.
yeah, it'll be very interesting.
And then just to follow up, the whole permutation entropy makes me think of density estimation, like kernel density estimation in particular, and it's just that the alphabets are different.
Yep.
Right? Because you want to have a probability over what you're seeing.
That's right. So one of the things that was shown in the Garland paper and also in a couple of earlier papers on permutation entropy is that if we can define a probability distribution, it will match the KL divergence from these distributions in time, at least the change in the permutation entropy. It will match the entropy of those distributions. It will also match, like if we can calculate, for systems where we can calculate a Lyapunov exponent, it will match the exponents. If we could actually even define like a Lyapunov function that bounds the stability, it's going to tell us in what scales we're going to be able to do predictions. So it's one of the really cool things about, it's a small literature on permutation entropy, but the papers that are out there are really awesome. I mean, they really have done a thorough job of demonstrating that, at least for systems where we know how they should behave, that this thing is telling us that that's how they behave. Yeah, I turned into, I'll use the word bloodbath because that's what happened. The first time I gave this talk, I assumed I was the only one that hadn't heard of permutation entropy before, because it's often the case that we're in these interdisciplinary fields where the first one to learn about something that a lot of other people knew. Turns out that like kind of nobody knew what permutation entropy was, at least in terms of some of the details that I'd really glossed over. But it's been interesting to dig into that literature. I suspect it'll start to increase as we see these bigger and bigger data sets. So the other thing I'll just say real quickly on that is you get a stable estimate of, well, the papers say you get a stable estimate of the permutation entropy in log d factorial time series, where d is the dimension. Our experience is it's more like d factorial and not log, but either way, it's much shorter than you would need for doing something. Even if you could do a Lyapunov exponent style approach, it's a much shorter time series. This also doesn't make any assumptions about stochasticity, which I know there are some new papers on Lyapunov stability and stochastic systems, but at least the classical stuff makes you come down on one side of the philosophical argument about the universe being stochastic or not.
I also hadn't heard of, or haven't been familiar with permutation entropy, so this might be like a completely
I bet it's
wrong
not.
question.
Yeah,
But I like, he said that the KL divergence kind of blows up when you see more of the data, and I'm wondering if there's other types of systems that, or spreading might be involved that like kind of bound the predictability of what you can do with that. So in science, for example, like if I have an uncertain next field that's going to emerge, more data might make me even more uncertain.
Yeah, so it's a good question, which is, are there other systems where we see this happening, in particular systems where we can make a prediction, like where we see these kind of spreading behaviors, where we think maybe there's social network differences. We haven't found them yet. So we tried this with the stock market data, mostly because we wanted to see if we could get rich, and what we showed is that the stock market's unpredictable in all dimensions, for the most part. We've done some creative things, and we think we found like a sliver of something in there, but the answer is kind of no. Apparently, this is kind of making me a little bit nervous. I thought like, here would be a great thing to do is I'll just download the rainfall data for Boston, and I'll show you what that looks like before we do this. And I've been pending in the queue on the US Climate Data website to download the rainfall data for Boston for the last 2 years for the last 72 hours, which has never happened before, so I don't know what's going on with that. I guess the Amazon Web Services were down, so maybe that's what's going on with it. But we would really like to look at, memes spreading on Twitter, the scientific ideas that you said, the emergence of new systems. So one thing I can tell you is that if we just train a Markov chain over these distributions, we don't get that increase, right? Like you have to see enough of the data that you realize the stable distribution of that chain, but it's not just a random walk process over a symbol distribution. It is if you don't have enough data, but after that, it's not. You actually have to change the distribution over which that chain is walking. So any system, they say if I have a really high dimensional system and I'm gonna regularize it in some ways, and I'm gonna change how it's regularized, that'll give you this behavior. If I have a hidden Markov process, that will also give us this kind of behavior. So those systems should all look like that. Two really fascinating things. One, if we train these Markov chains, all the best fit Markov chains have slightly higher entropy than the data. And what that we think means is that the real data has longer memory than we can pick up, which is kind of cool. The other thing that people have published on is that if you have a Markov chain with no hidden state, the correlation in time should decay exponentially, and we tend to see for real systems that it's a power law, which would tell us that there's some pathological hidden Markov process that's happening. But I think it's a really cool area of research that, like, when we talk about big data, that this big data thing kind of opens up our ability to look at these questions in a comparative way that we probably couldn't have even a few years ago.
Then can I just squeeze another one in there? Do you think it's more of what's driving this is more of the adaptive nature of the data that you're trying to feed in, or is it about just the model itself not being like?
Yeah, so we don't know. So the question is, what's really going on here? So we know that there's these social network differences, and we can show in simulations that we can get out consistent answers with what we see in the data. We also know that the model structure is changing, right? Like that's the reason we've got to use these dynamical modeling procedures. Just we have a vaccine, we don't have a vaccine, it evolves resistance to a drug that worked before, we stop washing your hands, we stop washing our hands, we run out of nurses, we run out of etc. We know that kind of thing happens. But we also know, at least not the rise in predictability, but the low predictability you get just out of a chaotic system where nothing changes. So one of the things that we're really trying to figure out how to push hard on now is, can we actually distinguish what's driving this signal? Because that to me, and it sounds like to you too, it feels like the really exciting question to push on. I would recommend not ever doing this, but I kind of pushed a little bit harder on the sort of cross-validation and these no free lunch ideas, and that was really not popular, right? Because one of the things that if you're trying to claim that cross-validation is not going to generalize, that's a big problem, sort of philosophically. So I would recommend not going down that path, except maybe over beers, but yeah.
So kind of continuing the kind of question. So like it may be good for one task, I mean, for some aspect, and maybe not so good for others. Is measure. So what other kind of measures you're thinking of predictability? What others you consider and what others considering for what task?
Yeah, so that's a good question. So, I mean, what other measures of predictability would we want to consider and for what other tasks, right? So, a measure of predictability that we would be very interested in is one that does not actually throw away the information about the magnitude, right? And we think we could probably accommodate that by defining a priori what the symbols look like. There are, of course, I mean, there's a huge body of literature from dynamical systems on how one would study the stability of these things, right? So for systems in which we can write down a system of equations and agree on them, we may be able, in a lot of those cases, to calculate what the Lyapunov exponent is, or in some of them they actually have defined like the full region of which you could bound the stability. In systems where we can define a probability distribution, we can lean on those existing methods related to sort of information theoretic properties in continuous space over those probability distributions. So I think in all of those settings, and in cases where we can kind of compare them, it's really useful. So the reason that we picked this tool off the shelf is for exactly this reason, that we didn't want to have to argue about the right model structure. But in cases where we think we have a good model structure, it's really fascinating. So I'm actually talking with a couple of researchers who are working on self-driving cars, and I'm very much a novice on this, so if anybody works on it, I'm going to mess it up. But my understanding is that the really hard part about the self-driving cars is the physics of the tire on the ground. The rest of it is kind of apparently very easy. But the physics of the tire on the ground is really hard. And so they're really working on these sort of detailed stability analyses of these PDEs that describe the wheel on the car in different kinds of conditions. And so we were kind of brainstorming about how one might take kind of a hybrid approach if there are questions that are slightly too hard to solve with some of these traditional ways. Could we convince ourselves that something like this would work in a kind of complementary fashion? But that's all sort of very new.
So when we talk about the data, is it, in your case, is it just time series data?
It's all time series.
It's all time. The thing is, I mean, for me, like the time series data is the output, and then there are the fundamental latent variables. And those ones you don't observe.
Yeah.
And if you don't know them, it will be noise, like in your case. I mean, for disease, you will see seasonalities, and I would say that's the most like time series can capture.
Yeah.
And all the other stuff will accumulate as noise, and that's why probably you see the increase of the entropy. I mean,
Yeah.
What if we know that the underlying, like, genetics, for example, the weather, like the things actually, the interactions, if we have those information, then I mean, I couldn't like see how you could improve like the predictability just based on the time series.
No, I don't think you can. Yeah. So the question is sort of what about all these things that we know are true about these systems, and is it really that surprising that we're seeing this? So I think, I mean, if you can measure these other quantities that we know are important, then it should improve our ability to do prediction. I think what this is very much telling us is that, for example, if I want to calculate the peak of influenza, trying to predict the full time series and then calculate the peak off of that is going to be much harder than just trying to come up with some model that generates a probability distribution over peak sizes, for example, because into that model I can feed all the things that you're mentioning. I think one of the things that we feel like is a contribution of this is that I think as a field we know that all those things are happening, but really trying to get a quantification of what that really looks like in terms of the data we think is new. And we think seeing these regularities across disease tells us that there's something about the biology or the sociology of these pathogens that's kind of uniting them, and maybe that means it can give us a window into what some of the bigger important drivers are for these systems, so that maybe we can kind of incorporate those into our models. But I agree with your sentiment very much. Yeah.
Okay.
I also, real quick, I will say too, of course, if we don't have any historical data, like if we want to predict the probability that Zika is going to show up in the United States, or the probability that Ebola is going to show up in London, like we don't obviously, we're not in this regime. We're in a regime where we have to have a model where we think we understand the physics and we can do forecasting from that, right? So I'm certainly not talking about those kinds of models either.
I cannot one more.
Yeah.
So it's related to something. So his question is kind of blocked on the following. So can you think of a maximum entropy time series subject to specific constraint? Right. So for example, it's not like there's some kind of hidden variables or not, but maybe some given out correlation. Right.
Yeah.
I don't know. So what would be the maximum time series, maximum unpredictable time series subject to this constraint? Can you think of this?
Unpredictable time series.
Yeah, so maximum. Yeah. So like, but they have this outer correlation. So you are considering a kind of an ensemble of all possible time series.
Yeah.
We all have.
And so you're meaning not one that we've rescaled to be one so we can compare across the time series, but I think it'll be really fascinating. Yeah.
I don't think this question is.
No, I know. So I mean, my understanding of this is that it's very, very hard to say anything mathematical about the entropy of these systems, right? So that's partly why we want to lean on this, because there are very, very smart people who I could never hope to be, who say that you can't do this. Right?
Just tell us.
Yeah.
Yeah.
No, so yeah, trust me, if I could define the entropy ball around these things in some sort of analytical way, I would have just put the equation up there and gone to lunch. But I agree with you that it's really fascinating.
Just one last question that is more a comment from you. So that's obviously give you some interval for prediction. There is good news, bad news, etc. Now there is a lot of approaches that works with novel digital data streams. Like, okay, you take all the time series of editing on Wikipedia, and then I do predictions for whatever diseases. I get the Google flu data, and all those approaches are based basically on linear regression, in which they throw in a bunch of data. There is the classic 80-20 things, etc. What do you think at this point in the light of this work of those approaches?
Yeah, so it's a good question, which is, well, I think there's sort of two questions. One is about the kind of novel data streams, and the other one is kind of about this sort of regression-based forecast. I think one, from the novel data streams perspective, I feel like this could be a really useful tool for justifying the utility of some of these data streams, right? So you ask this question like, how useful are the Wikipedia data? Well, we could maybe say something about how useful they are in the context of how they affect the predictability of some of these systems that we care about. From the sort of regression-based approaches, I'll say two things. One, I think that deciding that a regression-based approach is good feels like it has to be in the context of these information theoretic properties of the prediction, right? So if it's a system that's pretty easy to predict, maybe we're not that impressed at some of the regression forecasts that are good, because lots and lots of different models might lead us to good forecasts. I also think that in terms of the shifting sort of nature of the predictability in the time series, that these cross-validations training these linear models are really we're not getting any kind of guarantee that they're going to converge on the correct model, or even a good model. I think it's one of the reasons there's this very nice paper, at least I really love this paper by Cécile Laboud on using the method of analogs, which is the sort of vector-based forecasting from the weather, and that does really good for flu, like kind of 1-2 weeks out. I think sort of for this reason is that you build up a big enough distribution and you can kind of maybe find in the historical time series some bits that match your current bits pretty well. And I think often when we compete those against the regression-based forecasts really aren't doing very much better, sort of for kind of exactly these reasons. The other side note I'll say is, one thing we found is that like interpolating missing data, that kind of thing really leads to much higher entropy, because you're kind of inducing stuff that doesn't really exist before, right? So if I have a missing data point and I kind of do an average to fill it in, the entropy goes way up. So one of the hard parts is to figure out that we're convince ourselves we're getting the entropy right with missing data, which we did by simulation to show that we're not messing that part up. But I think it kind of tells us how we should imagine the cost and benefit of trying to do interpolation if we do have missing data. Like, we're going to use Google Flu Trends to fill in missing ILINet data. Maybe that's not necessarily as good of ideas to think about methods that might be robust to the missing data, like if we just have a reporting rate or something like that.
Okay, so thank you very much.